Deciphering academese. Yes, academic language can be obtuse, abstruse, and downright datal. For your convenience, we present a short thesaurus of common academic phrases. To the best of the author's knowledge, we were too lazy to do a real light literature search. Results were found through direct experimentation. We played around with it until it worked. The data agreed quite well with the predicted model. If you turn the page upside down and squint, it doesn't look too different. It should be noted that, okay, so my experiments weren't perfect. Are you happy now? These results suggest that if we take a huge leap of reasoning, we can get more mileage out of our data. Future work will focus on, yes, we know there's a big flaw, but we promise we'll get to it someday. Remains an open question. We have no clue either. So, the idea of confounders can be traced back to fertilizer tests done in the 1840s. Fertilizer tests in the Great Plains. And the idea of, we're going to compare this month to that month, but the fields are different and the weather was different. And we got to come up with some way to make the comparisons more comparable. In the 1920s, a fellow named Ronald Fisher was pushing the idea of taking your groups and what's called randomizing them, is that making each group kind of randomly comparable within certain parameters. And then you look at confounders. When we did the experiment design lab, is that we talked about you could do a design lab on driving with a hat or not a hat on. And nobody ever seemed to think about putting groups together, thinking in terms of age, thinking in terms of gender. Whereas if you had a bunch of older people in one of your two groups and a bunch of younger people in your other group, they're not going to necessarily be comparable. If you're skewed gender-wise and you're measuring speed in Northern American drivers, where there are gender differences, there's not all over the place, but there are in North America. Northern America, not Southern America. You're just likely to be run off the road by an older lady as a young man in down south. But mixing your groups so that they're comparable and taking into account that certain things need to be randomized, you can date back to the 1920s. And the idea of being able to do the thing over and over and over again, replicating, setting up a design that you could do over and over and over again. You start to see quantitative data, people using averages, which are called means, people finding a midpoint in the distribution of data, the median, which is not, it's often not the average, but it also can have some statistical importance. And the standard deviation, how much are your numbers ranging from what you're getting as an average? Is that there's ways to figure all that out. And it's all stuff that you need to get trained for, sort of. Is that an awful lot of modern experimental statistics is done with software. And I'm not entirely sure how acquainted the people that are writing these things down are with the actual math and what exactly standard deviation is giving them, what sort of patterns they're getting, statistical significance. Here's a big deal. What are the odds that your results are reliable results and not just the results of chancy distributions of numbers? There is a p-value that you get that compares your results versus the results if nothing really was working at all. It's supposed to be a comparison against the null hypothesis. Whereas you want a p-value of less than 0.05, although now they're debating is to make it less than 0.005. But it's become kind of the holy grail of, they'll accept your results if you can get your p-value under the required threshold. And people do what's called p-hacking, which is you play with the data. You can collect data a whole bunch of different ways, and then you can manipulate it a whole bunch of different ways. And you keep messing with it until you get a p-value that a reviewer is going to go, oh, okay, that's what we want. And a tremendous amount of published results don't give you all of the data. You can't necessarily go through their numbers reading an article and see where they got their p-value from. Sometimes you can, sometimes you can't. A big deal, this is more general. Just because two things go together doesn't mean doesn't mean they're really connected. Is that, the reason that people typically drive slower with hats on is that your typical population, depending on where you are, but a lot of times the people that are wearing hats are older, more cautious, more slow drivers. Wearing a hat doesn't make you drive slowly. It's a correlation, not a causation. And when you are doing your research or doing your observations and you see correlations and you can count the numbers and you get correlations, then it's a question of how do we figure out if there's a connection here or not? And if the connection is causative or not. Or they just two things happen to travel together, because older people drive slower. Here's a big deal right now. Bayesian analysis is building your current interpretation of data on things that you already know. I mean, to some extent, we do this all the time. One of the big tricky things about the modern, the two vaccines that are out right now, is that they were using a technology that nobody had been able to get to work in human vaccines. It had been around for 20-some years, a messenger RNA put into the cells to have them express an antigen. But no one had been able to pull off the actual application of it. Whereas some of the other stuff that's out there, the Johnson & Johnson is building unestablished technology on using a different type of virus as a kind of a ferry to pull aspects of the coronavirus into where your immune system can react to them. And that, even though it's taken longer to get that stuff rolling, is it the people that are doing the regulating and the reviewing were much more prepared to accept the early results from vaccine technologies that were well established for safety and effectiveness. Is it building on what was already known? Is that I'm still not entirely sure the messenger RNA vaccines are not going to wind up having some long-term effects that make sense, but we haven't really planned for. Again, building on what we know about how your immune system reacts to presented antigens, and the fact that nobody's telling us what cells are doing that in our bodies. Another thing that I've talked about before, and I talked about a couple of minutes ago, you get your results, you have evidence. You go, oh, this stuff supports my hypothesis. Is it this suggests that the hypothesis is right? Good science person doesn't say, oh, I've proven it. Sometimes you'll hear them say, I've disproven it because it didn't work. But even that's not entirely true. Sometimes it didn't work not because your idea was wrong, but because something else, some confounding factor was at work that you weren't aware was there. So even proof and disproof are two kind of taboo words in science. Is evidence is great, support is great, proof, because the thing is, no matter how much proof you think you have, there's still the possibility that your underlying hypothesis is wrong. You just keep, you're pushing in the right direction, but your explanation for what's really happening, you haven't hit the impediment that's going to go, oh, wait a minute, it doesn't actually work this way. Classic example being how gravity works, where it all seemed to make perfect sense as an attractive force between two objects. And then some things started to happen that didn't completely make sense. And Einstein came along and went, nope, doesn't really work that way. The math and all the support you've got is wonderful, but something else is going on here. Which is what I suspect, now, I don't want to get into that. Peer review, I've talked about this before, is that you're doing something very complicated and very specialized. In order for anybody to know whether you're doing it right, you kind of need other people who have experience in that subset of science to be able to look at your results. In industry, that could be the guy in the cubicle next door. In broader academic research, usually nobody else in your university is doing the same stuff you're doing. You may email with people that you know, but a lot of times you're in competition for funding, so you don't necessarily want to give away what you're doing. Postmodernism again. And a lot of times, it isn't until you've kind of got everything to what you think is work, and then you write up your results and you send them off to get published, and then they have to go through a peer review system, which can get a little dicey. But you can take years of doing research and have somebody in the peer review system go, no, there's nothing important here. We don't want to publish this. There's nothing new and interesting here. And of course, the result there isn't that you don't get published, it's that you don't get published there. Is that you eventually find a place to get published, because that's how the system works. I mentioned before, a lot of times people in the more rigid sciences they talk about hard sciences and soft sciences. Hard sciences are, physics and chemistry and biology. Soft sciences are more like education. And it's really just, prejudice, really. And I mean, you can support it with some of the, as I said, describing some of the way that they do their research. It doesn't really rise to the standard that you get in some of the other ones. But and now, just in biology, a hypothesis is a working idea that you're testing. A theory is an idea that not only has a decent amount of support, but people are kind of buying into. Is that so? There's a certain amount of culture acceptance, peer review acceptance for theories. Once something becomes kind of widely accepted, like, yeah, okay, we're pretty sure it works that way. Now, laws in physics are things that absolutely positively work a particular way, and if you find out they don't work that way, the law is supposed to go away. In biology, laws are a much fuzzier concept, and once something is called a law, it may stay called a law even long after the basic idea has been discarded. A lot of Mendel's laws of genetics are still called Mendel's laws of genetics, but turns out there's huge numbers of exceptions to them. They don't really work across the board the way that he thought they did. And people started to find that out pretty quickly, and they still call them Mendel's laws of genetics. Okay, so that's, again, a review of science with a little bit of more context for how modern science works. In a later chapter, we'll talk about science as a career and what the stepping stones are and how it all works, at least to some extent. We'll cover that later, though.